Hello, everyone, and welcome to another episode of Gen Live. I'm Juliana Lemire, science writer at Gen, and I'm joined today by two of my colleagues, Kevin Davies and Anjali Sarkar. Hi, Kevin. Hi, Anjali.
Hello.
Hi, Juliana.
On today's show, we'll be discussing the state of neuroscience research with three experts: Heather Dawes, the executive director of the Wheel Neurohub; Dietrich Steffens, CEO at Therapeutics; and John Van Leeuwen, the Global Brain Health Institute. We'll hear from them in just a moment.
Today's episode is sponsored by PerkinElmer. Expand the possibilities for single-cell transcriptional profiling with a Hive scRNA-seq solution. PerkinElmer and Honeycomb Biotechnologies recently launched the Hive scRNA-seq solution, which integrates sample storage and single-cell profiling into a complete workflow. The Hive scRNA-seq solution solves the issues that limit single-cell RNA analysis by enabling multi-site and multi-time point sample collection, maintaining sample integrity through storage, shipping, and processing, increasing the recovery rate of fragile cells, facilitating the loading of larger sample volumes, delivering flexible and scalable workflows through batch processing, removing the need for specialized instrumentation. The Hive scRNA-seq solution expands single-cell opportunities for basic translational, preclinical, and clinical research. Visit scRNA-seq.com to learn more about the Hive scRNA-seq solution.
Don't forget the hyphen. And Gen Live is also brought to you by Gen Biotechnology, the exciting new peer-reviewed journal from Gen, launching in 2022, publishing exceptional research across all areas of biotechnology. Manuscript submissions are now being accepted. More details at genbiotechjournal.com.
So neuroscience research has enjoyed robust funding for many years. I remember I was a technician in the lab, and when I was a technician in the lab, in the neuroscience lab, and the 1990s were the decade of the brain. And we're now beginning to see so much of that money was put to fascinating and diverse uses. Moreover, after receding from much of the neurodrug discovery realm for more than 2 decades, pharma has begun to express renewed interest in developing drugs to treat a whole host of neurological disorders. Novel therapies to treat diseases such as multiple sclerosis, Alzheimer's, Parkinson's, and muscular dystrophy are popping up alongside new biologics for migraines and gene therapy clinical trials for neurogenetic diseases like Huntington's. However, therapeutics are only part of the story, as much research has been devoted to creating comprehensive brain maps, from neuronal interactions of brain regions to the molecular multiomic patterns within specific brain localities. During normal and disease states, technology advances are truly making headway into one of the most enigmatic biological frontiers. So to discuss all of this today, we have 3 great guests. Heather Dawes is the executive director of the Wheel Neurohub. Heather works with leadership to chart strategic directions focused on achieving impacts in scientific understanding and clinical care. She's been at UCSF for almost a decade, previously working at Fidelity Foundations and in scientific publishing. In fact, she was a colleague of Kevin's at Cell Press 20 years ago. Thanks for joining us, Heather.
Thank you.
We're also joined today by Dietrich Steffens, CEO of Therapeutics, a Pittsburgh-based biotech developing a novel class of precision genetic medicines to address genetic diseases at the base level. Dietrich has helped launch 14 biotechnology companies to advance novel molecular diagnostics and was a co-founder of Navigenics, a pioneering consumer genetics company. Nubase is targeting several neurological disorders, including Huntington's and myotonic dystrophy. Thanks for joining us, Dietrich.
Thanks for having me.
And John Van Leeuwen is a neuroscientist who works at the intersection of science, medicine, and policy. He's currently the Director of Strategic Initiatives at the UCSF Wheel Institute for Neurosciences. His work focuses on research strategy, translational science, industry partnerships, and using technology to make a global impact. Hi, John, welcome.
Good to be here.
So we'll start today's conversation by hearing a bit about the work that John is doing. But before I pass it over to you, Anjali, I want to mention to our audience that our panelists will have time to address your questions as well. So please put your questions into the Q&A box at the bottom of your screen, not the chat. Okay, Anjali, over to you.
John, my question is for you. You work at the intersection of neuroscience, healthcare, and policy. Tell us a bit about how your journey to this intriguing junction came about.
Sure, happy to. And it's been, I think the word journey is a great word to describe it. So I started with an interest in neuroscience and was originally trained as a neuroscientist, a cellular molecular neuroscientist at one point in my life, but became interested in what really I realized what drove me was having an impact on people. So my research always was connected to disease and understanding disease, understanding health, and how to have an impact, and thinking about how my work can continue to have an impact. That led me to policy. I developed an interest in policy, and I did a policy fellowship in DC, worked at the State Department, the VA, and then eventually made my way back to neuroscience, realizing that as I was exploring the policy world and the impact that I was having there, it was fantastic and great learning experience, but it is such an exciting time for neuroscience, and finding an opportunity to get back to that sector was incredibly exciting. And so I've been at the Weil Institute in the role of Director of Strategic Initiatives for the last couple of years, and it's been a wonderful ride there so far. And one of the things we've done at the Weil Institute is thinking about how we can bring together the clinical and the basic neuroscience. So bringing together clinical departments, neurology, psychiatry, neurological surgery, and our strong basic neuroscience programs, and how we can have an impact in the translational space, as well as the basic discovery. And I'm happy to say more about the Weil Institute and some of the things that we've been working on, and some of what we're looking forward to over the years ahead. One of the things that Heather will talk about was the Weil Neurohub that came into formation recently, and what new partnerships that's been allowed to do. One of the things that I've been lucky enough to be involved with both is the formation of a new partnership with Roche and Genentech, what we call the Alliance for Therapies in Neuroscience. So it's been terrific working with colleagues in the industry side to see how we in academia can work with them in order to advance therapies collectively and have that impact on humans and have that impact on patients' lives.
In your vision statement on the Weil Institute website, you said that neurological disorders, including dementia, exert a global burden and necessitate global solutions. So what kind of global initiatives are being taken to address these neurological disorders?
Yeah, so I'll mention that I have a few different hats that I wear at UCSF. So one is with the Weil Institute for Neurosciences, where I lead strategic initiatives there. I'm also part of the faculty in the neurology department, and also the Global Brain Health Institute, part of the faculty for GBHI, Global Brain Health Institute, where I'm one of the leads for the policy curriculum there, and thinking about how we can have an impact. So a lot of my work on the global side is related to GBHI and the way that recognizing that diseases of aging, in particular dementia, are a devastating impact on the population, from all the countries that we come from, but also globally. Brain health issues, neurological psychiatric disorders are the second leading cause of death worldwide and the leading cause of disability worldwide. So oftentimes we think about these issues and problems as conditions that we deal with. I'm sitting in San Francisco, some of us here in New York, but the impact really is truly a global impact, that people are impacted by these diseases. And so at UCSF, at the Weil Institute, a lot of the focus has been on the basic research and clinical side of things, with the belief that as we make advances in our understanding of these diseases, both in what the basic underlying causes are, then also identifying ways that we can treat and care for these conditions, that we have a possibility from that to also then expand and make this have a global impact. So a lot of initial focus is on some of the early stages and what we're doing here, but with a mind towards how we can have these advances be accessible and beneficial to people worldwide.
Right. We'll come back to you, John. For now, I'll pass it over to you, Juliana.
Okay, thanks, Anjali. So Heather, I am curious about the Neurohub. I don't know that much about it. I know it's very new, launched in 2019, I think. So can you tell us what it is and how it came about and what's going on there?
Thanks so much, Juliana. Yeah, Neurohub is relatively new and it is unique. So I'm happy to speak to some of its origins and objectives and structure. As you mentioned, the Wild Neurohub was launched in 2019. It is effectively an enabling research umbrella that brings together neuroscience research communities at UCSF, at UC Berkeley, and at the University of Washington. In many ways, it's leveraging a bit of regional advantage writ large, as well as, I think, sort of a powerful perspective that comes with research stemming and housed within big public universities. So we have, going to, I think, some of John's comments just a minute ago, a broad objective of advancing clinical research and translational research that's going to be impactful globally, as well as helping to support basic neuroscience research that has the potential for clinical translation. The Wild Neurohub's vision is really centered on leveraging the talents and the particular mix of clinical research and basic neuroscience strengths across those three institutions. We also have affiliate relationships with a couple of nearby national labs, Lawrence Livermore National Laboratory, as well as Lawrence Berkeley National Laboratory, as well as the Allen Institute up in Seattle. And I think that the breadth of activity and expertise that we can draw on across the universities and across those institutes is commensurate to the challenge that we have when we're looking at some of the most difficult clinical problems that we have in neuroscience. We've had decades of trials and tremendous investment, very necessary and appropriately significant given the burden that these disorders have on us as a society. But we really have a challenge at this point to convert on that potential and start to find solutions for these disorders. Joan and Sandy Weil enabled the kickoff and establishment of the Neurohub in 2019 with a generous gift of a little over $100 million over 10 years to allow for the establishment of a series of internal research programs that are now well underway that basically emphasize team science and multidisciplinary research. But there's also, as John mentioned, a strong emphasis on reaching out to establish partnerships with industry and other stakeholders to ensure that we make the most of the opportunity for the new research that's coming down the pike.
Okay, great. Yeah, I was going to ask, I mean, when you say bringing together, different people in the area, different researchers, clinical, basic, is it more like funding group projects, like you were talking about team science? Is it also like increasing collaboration or communication between those people? Like, what's a kind of practical example of how people are brought together?
Yeah, thanks for asking.
Under the Neurohub,
Yeah, that's right. And there are multiple dimensions of this for sure. And, that's one of the reasons to establish an umbrella like the Neurohub, as opposed to just, kind of a single focus, pilot research grant program. This is really about building community in a way that's going to be productive, fresh, and make work possible that wouldn't happen otherwise. So in terms of examples, I'll mention one of our programs is our so-called Pillar Project program, which brings together investigators into different pillar projects that are focused on, fast-moving and fundamental challenges and opportunities in the field, like neural engineering or optical imaging innovation. These are in many ways a combination of research projects, team research, but also capacity building, looking for ways to build up what's possible from a technological and methodological standpoint across our community, helping to transmit that new capability and knowledge to our trainees and our collaborators within and outside the Neurohub. And in each case, these pillars are oriented to tackling kind of a key technological bottleneck that if we can move the needle in terms of what's possible, we have a good shot of making some progress in key areas that will actually have, represent an innovative advance and an impact on patients. So whether it's in the neural engineering realm and finding a way to establish closed loop neuromodulation algorithms for stroke recovery, or whether it's in, say, the deep phenotyping realm for patients and being able to establish capacity and structure for really bringing depth of data into our analyses to understand complex disorders like Alzheimer's better. These are sort of these dual capacity building and research projects that we really see as being potentially impactful.
Okay, terrific. And before I hand it over to Kevin and Dietrich, I want to ask you about the partnership with Genentech and Roche. So why, specifically, I guess, for neuroscience, why is partnering so important? And what will this particular partnership bring to the Neurohub? What will Genentech and Roche bring to the Neurohub? And what will the Neurohub bring to them?
It's a great question. And the partnership is something we're really excited about. I'll just say a few high-level words, and then I'll actually pass it over to John, who's been really involved in that partnership and helped make it possible. But I think just generally speaking, part of what is so exciting about this kind of partnership is the opportunity to bridge some divides that sort of organically arise in our field with respect to how different groups of scientists are incentivized and what they, are able to focus on and pay attention to in their work. In the academic realm, we have, faculty who, are able to move forward on specific questions that they're able to ask and answer, but don't necessarily have the insights and the mindset of really practically minded folks in industry who are forced every day to ask, why are we doing this? And I'll have to justify this to 20 different people at a meeting next week. And of course, on the other hand, on the industry side, there are limitations to the pipeline and thinking that represents a synthesis across fields that academic groups are able to bring to discussion. So I think that just in terms of making the most of the collective knowledge that we have across the field that sometimes is not easily shareable or convertible to innovative projects, this kind of partnership helps to overcome that barrier. And I'll just pass it over to John to say more about the partnership itself. John?
Yeah, happy to say a bit more. Picking up on Heather's comments, and the partnership is so important throughout all of what we've been doing at the Weill Institute and Weill Neurohub. So one way to think about it is Weill Institute was founded crossing the silos of different domains and departments to bring together neurosciences collectively together. And then with the foundation of establishment of Neurohub, it was extending those partnerships outside of one university and bringing together several universities. And then from Neurohub, we were able to have new partnerships, realizing that academic institutions have areas that they excel at, but when trying to have an impact on patients, there's limitations that we as academics also have, and that we know that we need to work with our industry partners to ultimately translate our discoveries into therapies that can impact patients. And so all throughout, we've thought about how we can have partnerships that can advance what the ultimate aims that we're moving towards. And the partnership with Roche and Genentech, what we've dubbed the ATN, has been fantastic. We have shared vision of making an impact for patients. And one of the things you mentioned at the start of the program was how there's been a retreat among big pharma in the neuroscience space, and some are entering back in. Roche and Genentech have been wonderful to work with. They've been stalwarts in the neuroscience space. They have long tradition in neuroscience. One of the things that's unique about this partnership for us is that it's a long-term, over a 10-year partnership is how it's originally envisioned, realizing that neuroscience is a challenging space and it's tough problems that we're trying to solve, and it takes time to have an impact in these areas. And I'd say what we bring together is the collaborative aspect of it. So we're complementing each other's strengths throughout. There's new discoveries, new ideas that we bring, and tools and ideas, but then there's also the know-how that Roche and Genentech have, and the scale that they're able to do things on, that collectively we're able to do more together. And it really is a very collaborative effort from top to bottom, from leadership to management to the scientific teams that are working on these problems.
Okay, thanks so much, John. I'm going to pass it over to Kevin now, but before I do, I just want to remind our audience to please put any of your questions for our panelists into the Q&A box at the bottom of your screen. Okay, Kevin, over to you.
Thank you, Juliana. Dietrich, great to have you on Gen Live. I've followed your career very closely over the last 15 years or so. You were a co-founder at Neurogenetic, as we mentioned in the intro, a pioneering consumer genetics company. And I remember vividly your vision was instrumental in the creation of the New York Genome Center. That's a whole other story. So you've done great things both in academia and on the commercial side. And recently you've decided to go back to industry to take the helm of Newbase, a publicly traded drug discovery company. We'd love to hear a little bit about your company's mission and how you think you're going to achieve it.
Well, thanks, Kevin and Juliana and Anjali for having me. It's a real honor and pleasure. I come at trying to understand the brain and what happens when things go awry from a genetics perspective. Each of us originates as a diploid genome in a little lipid bilayer, and our genes control our normal development, and misspellings cause a wide variety of every human disease. Every human disease is genetic. And so whether it's single gene disorders, whether it's cancers, whether it's complex genetic diseases, the list goes on. And so even brain diseases are all genetically driven. And so there was a period in my life where the goal was to catalog variants that seemed to be enriched in individuals with a wide variety of brain diseases. And once we populated those variant databases, the goal was to deliver that information to patients and their doctors so that they were armed, at least with a knowledge of what was going awry, even in the absence of being able to do something about it. And so Newbase is really the capstone on that work and is meant to be able to drug the genome directly. And we've got a technology, which I'm happy to talk about if there's interest, that is synthetic genetic medicines that can engage with a diploid genome and address gain of function mutations, address loss of function mutations, and even do high fidelity genome editing to address change of function mutations. And so the genesis of Newbase was really looking at the nascent yet very fragmented genetic medicines industry, which of course arose about a decade ago, and it's largely focused on drugging RNA at this point in time to do gene silencing and gene replacement, gene therapy. And now, of course, we have gene editing. And understand, so the punchline is we have wonderful proof of concept in humans in terms of each of those causal mechanisms that drive all human disease. And yet this first generation of genetic medicines companies are limited. So they're limited in biodistribution. So we do a great job at delivering medicine systemically and curing liver diseases. So how do we get to other targets, especially the brain?
Yeah.
Number two is,
No,
well tolerated. Tolerability, right? I mean, many of these medicines are immunogenic and cause toxicities. And so how do we engineer that out of a platform that can drug the genome?
Right.
How do we get precision to actually see even the smallest mutations in a misbehaving gene versus the wild type gene, which is generally essential for life, or other highly homologous sequences. And finally, how do we make these in a scalable way as opposed to investing hundreds of millions of dollars in factories? And we've all seen sort of the issue in not being thoughtful about that up front. And so we believe we've solved all of that with NewBase. And again, happy to go into details. If
I think Anjali will sneak in a question about the technology. But for those who haven't followed NewBase, Dietrich, how much of your pipeline is focused on neuroscience? And you did touch on the delivery, the thorny delivery question. And how are you, what's the current strategy for delivering your genomic medicines to the right targets?
Yeah, I mean, so we believe we have an unlimited opportunity to impact once we fully crack the code on the platform, which we're on the cusp of. Half of our pipeline is currently focused on diseases that have a CNS component to them. Huntington's, obviously, it's often missed that Huntington's also has systemic implications, whole body implications. And then myotonic dystrophy type 1, which is often recognized as a neuromuscular disease, has a CNS component, including cognitive deficits that patients complain about. So delivery, our genetic medicines are neutral in charge, which allows them to slip past the liver without activating the scavenger receptor system that seems to soak up these negatively charged genetic medicines, whether they're mRNAs or ASOs or siRNAs, and that's why they all go to the liver. And so we slip past. And then we spent a lot of time engineering a shuttle that would allow these compounds to get across the plasma membranes of any tissue in the human body, including the cerebral vasculature, and dump into the parenchymal space and get into the cell bodies of neurons. And we've been pleased that we can deliver these drugs subcutaneously. They're relatively large, 7.5 kilodaltons, so they exceed Lipinski's size exclusions, and get them into the brain in a nice, evenly diffused manner and actually engage nucleic acid targets. So we're excited about the implications to modulate genes in the CNS.
That's great. Anjali, I'm going to let you squeeze in a quick couple of questions for Dietrich, and then we'll move it to the general interest round.
Sure. So Dietrich, NewBase is using a proprietary drug discovery platform that you were just mentioning to Kevin, the peptide nucleic acid antisense oligonucleotide platform, I believe, called Patrol for short. How is this platform being used to develop drugs for neurological disorders?
Yeah, so visualize a single-stranded oligonucleotide with a backbone and nucleobases arranged in a particular sequence that complements a misbehaving gene of interest. And so that's the conceptual structure of what our drugs look like. The secret sauce here is that we've replaced a natural sugar phosphate backbone, or the versions of that have been derivatized to be stable, with a neutral water-soluble synthetic backbone. And the nucleobases, the natural nucleobases, have similarly been re-engineered and reimagined to be able to not only invade the human genome and pry apart the double helix and engage in Watson-Crick base pairing with single base selectivity, but these also have the ability to scan the outside of the double-stranded genome in the major groove and peer into the interior of the double helix using what are known as Hookstein binders. And once they find a complementary sequence through that mechanism, engage in triplex binding. And so, we have innovations on nucleobases that allow us to engage the human genome at any locus with exquisite selectivity and muck up the works, if you will. So what does that mean? That means that we can, just using these oligos, we can do jujitsu moves on the machinery that runs on the rails of the genome. So for example, we can put a lock on the 5' end of a gene and inhibit RNA polymerase from being able to transcribe that. Therethrough completely shut down downstream protein production in the case of a toxic gain of function mutation. And similarly, we can open up regions of the genome so they're more accessible to the machinery and increase protein output. And the final trick is being able to, again, open up the double helix and insert a DNA donor strand, which can engage on top of a mutation that triggers the endogenous cellular machinery to come in and edit that mutation out. So we're not putting in, for example, CRISPR-Cas9, we're tricking the cell into thinking it's correcting its own mutation. And so that's how this stuff works. And there's a lot of chemistry in the background, which I won't bore you with, but we're quite excited because that seems like the final frontier in creating genetic medicines, is directly addressing root causality.
In addition to single gene diseases like Huntington that you just mentioned, we have complex neurological diseases, and you've played a significant role in unraveling autism as well. So what are the current challenges in preventing the development of drugs against these multifactorial, polygenic, multi-threshold neurological as well as neuropsychiatric disorders?
Oh my goodness, that's a hard question. So yeah, there was a point where we were heavily funded by the NIH. In fact, 15 NIH institutes banded together in the 2000s into what was called the NIH Neuroscience Blueprint. And they funded a genomics consortium that I had the privilege of leading that included Yale and Duke and Stanford and UCLA and my center. And we performed at scale genomics scanning projects, one of which was the Autism Genome Project, which was samples collected from affecteds and their family members across 50 clinical sites worldwide. 10,000 plus samples were scanned, and that contributed significantly to the understanding of copy number changes in individuals with autism. And we found a couple of smaller mutations such as the CNTNAP2 gene and so forth. So yes, we found a lot of genes. I think the issue is that we still don't understand how they all interact and where the central nodes are that are the high impact therapeutic targets. Short of that, we can't develop medicines unless we know what to drug. And so in these networks, it's, at least to me, it's been very difficult to say, aha, if we drug those three things in this way, we'll get the desired output. And so I feel like we as a field are still waiting for that in some of these complex genetic diseases.
Thank you. I'll pass it over to you, Juliana, now to get us started by asking some big picture questions.
Oh, sure. I actually want to go back to the Neurohub for 1 second. So what I'm wondering, because it is such a unique program or collaborative effort, I'm wondering, if you think Heather and John and Dietrich, do you think neuroscience as a field where pulling in multiple stakeholders is of particular importance? Or do you think that, any field, if we talked about cancer or anything else, could benefit from something like the Neurohub? Or is there something about neuroscience specifically that really makes it, super relevant?
Yeah, that's a really interesting question, Juliana. I think just the nature of the complexity of biology means that two heads are better than one, generally speaking, when you're trying to tackle a tough challenge. But I think that the nature of those challenges in neuroscience is arguably more complex, in part because of the dynamic nature of the nervous system over development, over the time span of disease progression. There are just many dimensions that need to be taken into account and understood. And I think that Dietrich's description of sort of, the challenges that we're left with this rich set of genes for autism and the incredible outputs of the Blueprint effort, typifies the reason why bringing multiple perspectives and research stakeholders is really an imperative, given the challenge that we have for making progress on complex questions. So Yeah, I would argue that neuroscience is, particularly well suited or well positioned to benefit from this kind of approach, but certainly not unique. I don't know if John or Stefan has anything, or Dietrich has anything that they want to add to that, but that's my perspective.
I think the way you ended it there, Heather, was perfect, that it's certainly well suited, but not unique. I think it speaks to where science is heading. There's this convergence of so many fields on these problems that I think any discipline now, you need to have computational and data science, you need to have engineers, you need to have chemists. I mean, there's a whole litany of specialties that are needed to have a big impact. But as you said, Heather, I think neuroscience in many ways is inherently multidisciplinary, and so perhaps more so than other fields, but certainly not the only field that necessitates this approach.
And I'll just add that I think that bridging the divide between, basic science and human biology is particularly challenging for neuroscience. It's another reason why, these academic industry partnerships are potentially very useful, because, and again, not unique. We all know that cancer has, had many, many disappointments when it comes to preclinical models translating. But I think that we're also very aware of just how many failed trials there have been in neuroscience, particularly in the domain of neurodegenerative disease. So, I think that divide is one that we have an obligation to try and cross.
Mm-hmm. Dietrich, you were saying earlier how you worked on a project that was really heavily, genomics based. And it seems to me, as somebody who writes about neuroscience, that now there's so much work being done on building maps and mapping the brain, and how mapping the brain is going to, in the connectome and the interactome, and how this is going to really, hopefully, increase our, as Heather was saying, our basic understanding of the brain. What role do you think, as somebody who's seen this, over the last couple of decades, from genomics now to maps to, who knows, maybe something else in the next decade? I mean, what kind of a role are maps going to play, do you think, in opening all of this up?
Let me preface my comments by saying that, the human body is the most complex machine we'll probably ever encounter in the universe, and the brain is the most complex, or I would posit the most complex organ in the human body. The more I learn about the workings of the brain, the more truly staggering the dimensionality is, and the depth and breadth of the processes that operate, and how, at least, the way I think about it, we're just at the tip of the iceberg at understanding any of those processes. So with that context, it's difficult for me to envision the form that a map would take. I mean, we've done structural analysis at a gross level, and maybe at medium scale level, we understand the genes and how they're turned on and off in neuronal populations and what happens when they go awry. But the intermediate, or the information that connects all of that it's difficult for me to understand how we capture that and integrate it into a map that we can actually engage with. And so I'm sorry you've reached the limit of how I can be helpful on this topic, but maybe my colleagues have a better path forward for creating the understanding of how the brain and the mind work.
I'll just add that maps are essential, but necessary but insufficient in some ways, I would say, given the complexity of the nervous system, and to the extent they can be used to develop sort of multi-scalar, multi-dimensional, sort of systems-based hypotheses for how the nervous system works under different circumstances, I think might be a productive way to think about maps, and kind of going from 2D and 3D to 4D and thinking about dynamic systems. And one thing that we're not always challenged to do in neuroscience is really think about this interaction across the different scales that we as individual researchers are accustomed to working in. So if we can kind of challenge ourselves as a field to build our maps, but then ask, how do these maps effectively work together to generate processes, including development and disease processes, I think we'll start to scratch the surface on the complexity we need to confront to make progress. It's a tall order, but I think, we're fooling ourselves if we think it's simpler than that.
Okay, terrific. And before I pass it over to Anjali, who I know has a couple more questions, I want to get to a great question from an audience member. And they asked, how much has genetic specific iPSC lines and 3D culture models impacted your space thus far? What do you think are the main obstacles for enhanced brain models really improving drug discovery and drug testing in the future? So I guess anybody who would like to take that is welcome to.
I could try. So I think brain organoids are fascinating and could be useful for medium throughput screening. But I don't think that they're fully representative, and that the desired outputs for most diseases can be measured in those systems. And so I'm still struggling for how we incorporate them, for example, into our drug development pipeline as a useful tool above and beyond what we can see in, for example, transgenics and other higher order species before we get to humans. So, yeah.
Anything to add, Heather or John, to that question about models?
I think from my perspective anyway, just to kind of echo, the opportunity we have to better understand and define how most effectively to use organoid cultures, induced pluripotent stem cells, other tissue-derived or engineered tissues, figuring out how to use them intelligently and keep that right balance of opportunity and possibility in mind on the one hand, and remaining critical about the limitations at the same time, I think is the key.
Mm-hmm.
Yeah, I'd agree with Heather and add that, as Dietrich was saying, it's still early on and we're still figuring out what the potential of these tools are. But I think there is exciting potential. One of the challenges that we always have, and that came up earlier in the conversation, is trying to understand what's relevant to the human, and the ability to have these be human-derived tissues. There's another dimension that it adds, but certainly not capturing the full complexity of an intact human.
Juliana, may I add something? I fear I'm coming off as fatalistic. So in the absence of deeply understanding the brain via all of the great work that's being happened, there's still incredible progress we can make in developing CNS-based therapeutics simply by measuring sort of inputs and outputs and not really fully understanding what's happening in the middle. And so a broken gene equals this disease. These 3 broken genes equals this disease. And you can start to piece together small areas of understanding that you can then throw medicines at. And so all is not lost, and there's lots more to figure out.
Yeah, that's a really great point, Dietrich, right? Well, we don't need to have the complete comprehensive understanding of the brain in order to make progress. I think I really like that you made that point. Anjali, I'm going to pass it over to you now.
Sure. On the tail end of what you just mentioned, Dietrich, that it's a complex picture, and we just need to figure out the targetable, druggable part that we can use in treatments. But and you mentioned earlier contactin AP2 and the other several genes that have been discovered. how do you think the field of neuroscience is using concepts such as multi-drug treatments from other areas such as oncology in the treatment of neurological disorders?
It's a wonderful question. I mean, I think that there, at least for us, there were some really interesting examples of taking off-label or on-market drugs for other indications and basically mapping them against new genes that we had identified. And so, you know, in that New England Journal of Medicine paper where we identified the CNTNAP2 gene in a Amish family with Holmes Morton in Lancaster, Pennsylvania, big, huge family, like clockwork, kids got these severe symptoms at 18 months of age, you know, regression of IQ, loss of language skills, went into status epilepticus. We were able to, in the lowest generation, identify kids that had not yet converted, but who were mutation positive and would at 18 months, like clockwork, get the autistic phenotype. And in those kids, pre-symptomatically, we were able to give them a generic anti-seizure medication in the hopes of simply trying to proactively control the seizures. And years later, those kids had not converted to the phenotype. And while it's not specifically a cancer to CNS sort of map that you had asked about, I think generally it's an example of trying to get better informed by genetic targets, whether it's early diagnosis or a new pathway, and map existing drugs against them. So I think there's great promise in doing that. And we should always look at those molecules first because they've been through the ringer already and have arrived on market, and not try and reinvent the wheel every time.
Repurposing is definitely key. So in addition to that, from a big picture point of view, what is the difference? What are some of the differences in developing drugs for neurological disorders as well as neuropsychiatric disorders versus the typical drug development and drug discovery pipelines? And this is for all three of you, whoever wants to pick it up.
Definitely defer to Dietrich on this one.
Well, I can start, and then I'm sure my colleagues have other great advice for everyone. So first of all, it's getting across the blood-brain barrier, and so it's been one of the grand challenges of drug delivery to get across this very tight filter that the human body has put in place to protect the delicate brain from all of the gunk that's floating around in your blood. And historically, there were some rules emerged that are named Lipinski's Rules of Five that define the characteristics of what a molecule should look like to be able to cross the blood-brain barrier. And they had features such as molecular weight, so things lower than 500 Daltons diffuse through the BBB, and generally things above 500 Daltons don't, and so forth. So that has been the traditional number one, I think, barrier. And you only start to understand that once you get into in vivo systems and higher order animals, recognizing that often mice have permeable blood-brain barriers. And so you have to get up into NHPs. And by that time, you spent a lot of time and money. I think the other is threading the therapeutic index gradient. And so any drug that you'll take orally or IV will go to the liver and kidneys first and achieve high concentrations there before you get therapeutic concentrations in the brain. And so that's a whole nother hurdle, is threading the needle between a toxic dose and effective dose. And then, of course, there's all of the phenotyping and sort of clinical endpoints that become nuanced and esoteric in brain diseases, below the low-hanging fruit. And just think about all the effort that went into creating surrogate markers, such as PET imaging, to more rapidly test Alzheimer's disease drugs, so you wouldn't have to do a 20-year trial in thousands of patients to get a signal as to whether you're slowing the progression of disease. And so the endpoints is another issue. It's hard. I think we're, as a field, getting much better at it. But those are three.
Looking forward into the future, what areas of neuroscience research do you think will see the most progress in the coming decade? John, shall we begin with you?
Sure, happy to start. And I might pick up actually where Dietrich left off and say that, you know, the endpoints and monitoring disease has been a real challenge for the neurosciences. And I'm very optimistic about new ways and new tools to monitor. Blood-based assays, I think, can be tremendously helpful. And there's been a lot of progress in this space. And so there's a range of disease that I think, you know, Alzheimer's being one of them, where this will open up new possibilities for how we conduct trials, how we monitor patients. So that's one that I'll mention that I think is really exciting. Computational is the computational side of things for neuroscience, I think will continue to make an impact. We're learning a lot from neuroscience is informing development of AI, and AI is informing how we approach neuroscience. So I'm curious to see where these things go. And I don't want to, I think sometimes there's a lot of hype with these things. So I'm interested to see where this goes, but I don't think it's going to solve all the problems that we have when it comes to the complexities of biology and the brain. And I think also the last one I'll mention is and something that Dietrich knows quite well, you know, the molecular and cellular therapeutics space is very exciting right now, from CRISPR, of course, but also cell-based therapies, the things that we've learned from cancer and how they're advancing that space. I'm very excited to see where this goes as we try to bring it over to neuroscience.
Heather, would you have something to add to this question about the future areas of research?
Well, certainly adding to John's excellent points, and also just to pick up a little bit on your earlier question too. I think one of the key challenges with developing drugs for neuroscience actually relates to understanding the underlying heterogeneity of disorders and disease, something that is particularly challenging in neuroscience because we effectively, in most cases, need to do that in either in a non-invasive way or through peripheral markers, as well as behavioral markers. So I think linked to that, I'm optimistic about this being a key area of advance over the next 10 years in neuroscience, is getting at patient stratification, understanding underlying heterogeneity for complex disorders like Alzheimer's, like major depression. And through that better understanding, being able to design more effective exploratory early stage trials and get over some of the humps that we've had in the past in developing drugs and therapies for neuroscience, because we haven't been able to either fully appreciate the underlying heterogeneity of these disorders or assess them in patients and be able to, break down subpopulations that are more likely to respond to our approach than others.
And if I may ask about funding, Heather, neuroscience requires a ton of money, and from your perspective, is the funding in neuroscience research enough, and is it being effectively used compared to the budgets we have?
Yeah, well, that's certainly a loaded question that I'm hesitant to answer head on. But I will say, I have been and I continue to, work on the funding side of research. I actually continue to advise for the Fidelity Foundation. And I think that, going back to an earlier comment that I think I made, I think this investment has certainly been commensurate to the challenge and obligation we have to make progress and solve these problems because of the burden on society. I think that one of the really encouraging things that helps to ensure that this investment is going to be utilized as effectively as possible is the evolution of how we approach research. And a continuing effort to think critically about what's working and not working, and to think creatively about bringing expertise together, updating how we do training to help ensure that we stay kind of at that forefront of knowledge generation, knowledge synthesis. So the Neurohub is one example of kind of innovative community building in neuroscience, but there are many others. You'll see trends, at least in terms of on the philanthropy side, in terms of building consortia together, where groups of interdisciplinary researchers are obligated to share data and findings before publication with one another to effectively take the laboratory, the classic academic laboratory model, and scale it up so that there is more free-flowing information. I think that's another example of innovation on this front. I think a lot of it has to do with innovation on the research community side, innovating in terms of data sharing, and also encouraging people to think differently about the challenges, the questions that they're asking. And in particular, there's a great opportunity to continue to update the exposures that our trainees have and encourage them to think more broadly about their science and the clinical implications of what they're doing. So I'll leave it there.
Okay.
Yeah. Can I get in a quick
a
corresponding question about investment in industry? Dietrich, we go through periods where many big pharmas just seem to abandon neuroscience as a vertical because it's too difficult. And then we get into periods, maybe we're in one now, where we're seeing some really innovative new technologies and biotech companies such as Newbase get off the ground and show promise. I wonder, from a 30,000-foot level, where would you say we are on that sort of, in that pendulum?
It's interesting. It's absolutely cyclic, and oftentimes it's driven, the down cycles are driven by a notable failure by big pharma. So for example, when Biogen's Alzheimer's antibody first failed, everyone ran away from Alzheimer's. You couldn't do anything you couldn't get anything funded in Alzheimer's disease. Yep. And then it's back, and suddenly that's a viable. Same with Roche's Huntington's disease phase 3 failure. We got penalized for that because Huntington's was out of vogue, and it still hasn't come back yet. And so, this herd mentality is so silly because there's so much dazzling innovation out there in the marketplace that it's unfair to draw lines anymore, I think, just based on a particular indication. And so I hope that investors who fund companies can take the time that it takes to really suspend disbelief and engage with the science until they deeply understand it. Maybe that's simply not feasible in this world we're living in, but I think that's what it's going to take all the way around, is being thoughtful.
Yeah. Heather or John, you have a comment on that? Okay. Dietrich, in 30 seconds, we had an audience question just asking you, you touched on your technology earlier, as well as the better known gene editing technologies. And I think the question in a nutshell was what sets Newbase's platform apart from the other areas where we're seeing great progress in precision genome engineering, base editing, and now prime editing, and so on.
Yeah, real quick. So for in vivo editing, you need to deliver a big honking protein that's highly antigenic, and generally they're going to go to the liver, much like the Intellia drug did. So what we have is a very small non-immunogenic series of oligonucleotides that can pry open the genome, insert a donor strand in, and recruit endogenous cellular machinery to fix the mistake. And so that can be delivered anywhere in the body, and it's non-immunogenic. And so that's the difference. The other is that we don't induce double-stranded breaks, which I think can be cytotoxic or cause damage. These are mismatch repair enzymes predominantly that do the work.
Very good. Thank you so much, Juliana.
That's okay. I had a question for John about strategic partnerships, but I think we're going to have you back on, John, because I think we're going to have to run. But anyway, so I think that brings us to the end of this session of Gen Live. I want to again thank John, Heather, and Dietrich for a terrific discussion, and we look forward to following all of your work in this area. Thanks, Anjali, Kevin, and the rest of the Gen production team for making this episode run so smoothly. And thanks again to PerkinElmer, our sponsor. And thanks also to all of you for tuning in. Don't miss our last Gen Live, the last Gen Live of 2021, which is next month. And we'll be discussing COVID-19, where we started our Gen Live show, with guests including Greg Zuckerman, who just wrote a new book on the development of mRNA vaccines. The book is called A Shot to Save the World. With that, I'm Juliana Lemire. Stay safe, and we'll see you next month.
Thanks, everyone.
Bye-bye,
Great
everyone.
to see you.
Thank you.
Bye-bye.
Goodbye.
Thanks all.